Please welcome our next speaker.
Hello everybody, my name is Kris Janssen. I work at Leuven University, Department of Chemistry. Next slide please. Classically or typically when we look at genetic disease, the picture that we get is very simple. So people think that okay, if an alteration or an error gets introduced in our genetic material, genes change, and as a result also the function of proteins changes, and this induces disease. But this is actually not the only process that's going on in our bodies. We also have epigenetic factors to take into account, and we also have to look at the bacterial metagenome. Next slide, please. So what is epigenetic modification? Epigenetic modification is the introduction of small chemical groups into the genetic material. These can be methyl groups, hydroxymethyl groups. Long story short, these chemical modifications change or modulate the activity of genes. They either get suppressed or the activity gets augmented. And these epigenetic modifications happen in function of environmental factors, and they can be transient over time. They are implicated in very important disease processes such as cancer. Next slide. Bacterial metagenomics, on the other hand, they are related to the symbiotic species that every human carries around in their bodies or on their bodies. So we have fungi, viruses, but especially also bacteria in our body. And these bacteria have the power to actually influence the host in very significant ways. For instance, by the metabolites that they produce, they can influence the host immune system. And as such, they are implicated in many autoimmune diseases, but also very prevalent diseases like diabetes or obesity. Next slide, please. So if we actually have a way to reliably map or get a picture of the epigenetic landscape of a patient, or actually the composition of its bacterial metagenome, we could actually target cancer treatments in a much better way or come up with probiotic treatments for many diseases. How do we do that at KU Leuven in the Hopkins group? Well, we have a complementary set of enzymes and small molecule fluorescent cofactor labels, which we can use to sequence specifically or epigenetically specific, so target specific epigenetic markers. We can put the labels onto our DNA. Next slide, please. That results actually in what we call a fluorocode, a DNA fluorocode or a genetic barcode. So we can image that fluorescent barcode by actually using super resolution fluorescence microscopy. The barcode that comes out, next slide please. So that barcode that comes out, we can actually use to match against databases of known genetic information, and we can actually profile the epigenetic landscape or the bacterial metagenomic composition of a patient. Next slide, please. This gives us many benefits, chiefly among which our methods are single molecules, so there is no requirement for DNA amplification, no non-specific chemical treatment is needed, so we can, in a large degree, suppress sample bias and loss of accuracy. Thank you very much.